Thank you to Professor Orkin for accepting our invitation on behalf of IGIB and Professor Alok Srivastava. He's the director at Stem Cell Center for Stem Cell Research at CMC Vellore. So he will be sharing this session. So he will initially introduce the speaker, and then he will take over the session from now onwards. Professor Alok.
Thank you, Dr. Shiva Ramalingam. I want to start by congratulating you and the IGIB for putting together a series of lectures on hemoglobin biology and its impact on clinical medicine. We have a very important and interesting talk today. The speaker of today's talk is the David G. Nathan Distinguished Professor of Pediatrics at the Harvard Medical School, a member of the National Academy of Sciences and the Institute of Medicine, and a HHMI investigator. He's a Harvard Medical School alumnus, and his lab has been interested in the genetic basis of blood disorders. They investigate the development and function of the blood system, the relationship between cancer and stem cells, and the mechanisms responsible for self-renewal of stem cells and the switch from fetal to adult hemoglobin. His laboratory cloned the first hematopoietic transcription factor, GATA1, in 1989, and then used GWAS studies to identify several other SNPs that correlate with fetal hemoglobin expression. In fact, they showed that BCL11A functions as a silencer of the gamma globin gene, a fact that has now been exploited in many ways for clinical applications. Hemoglobin disorders are a major public health problem in the country, and there is immense interest both from basic scientists as well as clinicians in India. So it's a pleasure and privilege for me to introduce Professor Stuart Orkin to deliver his talk on turning on fetal hemoglobin for therapy, mechanism, and translation. Professor Orkin, the stage is yours.
Thank you. Can you see the slides? I hope you can see the slides.
Not yet. So I think you need to share your screen maybe.
I am.
It was there earlier. Yeah, now we're there.
Now I just want to be certain we can move to the next slide. Will that move?
Yes, it did.
Okay, good.
You are
All right. Thank you very much for the kind introduction. I'm glad to be here virtually. I'd rather be in person, but I think we can reach more people this way, in fact. And that's what we're learning in this pandemic times. What I'd like to do in the next 45 minutes is review really some of the background and where we've come in this search to turn on fetal hemoglobin, and really what remains to be done, I think, to really bring this to reduce the burden of disease in both thalassemia and sickle cell disease worldwide. Now, the problem I'm talking about today is really an old one, that is regulation of fetal hemoglobin. And I like this quote by the French novelist Marcel Proust, who said, "The only real voyage of discovery consists not in seeking new landscapes, but in having new eyes." That is, having new ways of looking at the problem. And as was just mentioned, the hemoglobinopathies really are a burden to health systems globally. And this is on the left, a review by the late David Weatherall, which really emphasized this point that the disorders of hemoglobin are really an emerging global health burden that are increasing rather than decreasing in their impact, and that there are more than 3,000 children born each year with a hemoglobinopathy. Most of these children born in low or middle income countries, as illustrated below. And in another study shown here on the right emphasized this point again, really that this is a increasing rather than decreasing problem. In the United States, we have about 75 to 100,000 individuals with sickle cell disease. Here, the two major hemoglobinopathies are summarized. On the left is beta thalassemia, a deficiency of beta chain production. And at the bottom are various mutations or places in the beta globin gene where mutations have been discovered that affect production of beta globin. These are concentrated in different geographic regions. So in different parts of the world, one would focus on different mutations. Most of those mutations we actually discovered back in the 1980s using emerging cloning methods. And this was work we performed with Haig Kazazian at Johns Hopkins. On the right is sickle cell anemia, which instead of a myriad of mutations is a single mutation at position 6 of the beta globin protein, which changes the properties so that the protein tends to sickle or polymerize. Sickle cell disease was first described more than 100 years ago, but it was called the first molecular disease by Linus Pauling in 1949. And then in 1956, Vernon Ingram discovered the amino acid substitution. And that's really a paradigm of early human genetics. Now, the role of fetal hemoglobin as a modifier of severity of the beta globin gene disorders has been recognized for many years. And in terms of sickle cell disease, one of the first recognitions was that of Janet Watson, a pediatrician, who in 1948 reported that babies who were destined to have sickle cell disease were not ill in the newborn or neonatal period. And she attributed that to their high fetal hemoglobin. And in fact, she was correct. And then a natural history study shown at the bottom, conducted in 1994, showed that the lifespan is longer in sickle cell disease patients who have high fetal hemoglobin. And the same is true of individuals who have beta thalassemia as well. The severity is inversely related to the level of fetal hemoglobin. So if one takes sickle cell disease as an example, but I think one could do the same for thalassemia, it's a single Mendelian disorder with a very variable clinical presentation. And in the case of sickle cell disease, where one wants to interrupt the sickling process, one could do that by interfering with polymerization, change the oxygen affinity curve, which has recently been attempted with a drug, Veloxetor, by Global Blood, or change the hemoglobin. And in modern genetics, that would be repairing the sequence or providing a new hemoglobin, that is fetal hemoglobin. And in thalassemia, where there's a deficiency of beta globin, one could add globin or replace the missing globin with fetal hemoglobin. Now, the most important sort of biology is shown here, which is the switch from fetal to adult hemoglobin. At the top is chromosome 11 on the short arm, where the locus control region enhancer element regulates the downstream genes, the embryonic to fetal genes in the adult beta globin gene within the cluster. And there's a switch from embryonic to fetal to beta chain during development as shown below. And as adults, we have about 1% fetal hemoglobin, which is largely restricted to a small population of cells called F cells. And really the therapeutic goal that's been envisioned for many years in the field is to increase that level from 1% to something on the order of 20% if it was distributed in all cells. And that would be sufficient to block the sickling process and have a substantial therapeutic benefit in thalassemia. If one could increase that even further, it would even be of more benefit. Now, there have been many attempts over the years to try to manipulate the level of fetal hemoglobin. This was an early attempt for nearly 40 years ago now at the NIH, where Tim Lay in Art Nienhuis's lab used the agent 5-azacitidine to try to increase fetal hemoglobin based on the hypothesis that methylation of DNA was preventing fetal hemoglobin expression. This had a positive result at a short term, but could never be turned into a real therapy. I should mention, for those of you who are familiar with Arthur Nienhuis, that unfortunately he just passed away earlier this week at the age of 79. A subsequent attempt to increase fetal hemoglobin was one that I participated in with Ora Platt and David Nathan at Children's Hospital in 1984, which was to use the agent hydroxyurea in an attempt to raise fetal hemoglobin, with the notion that changing the replication kinetics of progenitors might have some role. This does work, in fact, variably in patients with sickle cell disease, not really thalassemia. We do not understand the mechanism of action even today. However, this agent is approved for therapy in sickle cell disease and is a benefit to many patients. But really the questions that we've wanted to address over the years is, how is fetal hemoglobin actually regulated? That is, what's the switch? What's the mechanism? Can we use this knowledge or improved understanding for new therapies, genetic therapies, and what challenges remain? The breakthrough in the field came really in 2007, 2008 with the application of genome-wide association studies. An example is shown on the left, where one measures in the population hemoglobin F levels and correlates them with SNPs across the genome and tries to correlate them with genomic loci. The published studies are shown here on the right. The first one was by Swile Thien in Nature Genetics in 2007. And we collaborated with a group of the late Antonio Cao in Sardinia and with Joel Hirschhorn at the Children's Hospital. And the key feature of all of these GWAS studies is that there were really only 3 genomic loci identified in the so-called Manhattan plot. One on chromosome 11, which is the beta globin cluster, another one on the chromosome 6, which is the MIB oncogene, which affects maturation or development of cells. But the new locus was BCL11A on the short arm of chromosome 2, which is shown there under the Empire State Building. And if one sums the genetic contribution of these 3 loci, it's about 50% of the entire genetic variation of hemoglobin F. So this is an enormous genetic contribution to a trait for genome-wide association studies. And these SNPs are found throughout all populations. They're not restricted to populations that have a hemoglobinopathy. It's a common genetic variation. So really the challenge presented in GWAS is going from the entire genome of 3 billion base pairs to relevant targets and sequences that tell you something about the trait or something about a disease that is a few base pairs. And we've done that over about nearly a decade, where we've gone from genome-wide association to focusing on the BCL11A locus, focusing on an enhancer within the locus, finally using gene editing to really identify critical sequences. And these have been applied now in gene therapy and gene editing, as I'll come to later in the talk. The initial evidence that BCL11A had something to do with repressing fetal hemoglobin is summarized in an experiment here that was done by Vijay Sankaran, who was an MD PhD student in the lab at the time. And he used CD34 cells maturing along an erythroid pathway in culture. And we introduced into those cells shRNA for BCL11A, and one sees in the purple there a rise in gamma globin RNA expression with 2 different shRNAs or hairpins. And the other critical feature of BCL11A, which is shown on the left in a Western blot, is that BCL11A is really a definitive hematopoietic or adult-specific factor. It's not expressed in primitive yolk sac cells in human. It's expressed weakly in the fetal liver, and then it's expressed fully in adult cells in the bone marrow. And even in that, even after that experiment in 2008, we said in that paper that BCL11A emerges as a new therapeutic target for reactivation of hemoglobin F and sickle cell disease and the beta thalassemias. Probably the most critical genetic experiment we performed is this one, which was to ask how important is BCL11A as a single factor. And in this case, we used Berkeley sickle cell mice, which are mice engineered to express the human sickle beta gene. And these mice have no mouse globin genes, only human globin genes. And so they have a sickling phenotype. And we crossed these mice with conditional mice for BCL11A and knocked out BCL11A in erythroid cells only to restrict the study to those cells with erythropoietin receptor CRE. And really the question was, if we knocked out BCL11A in the erythroid cells, would we rescue the sickle cell disease phenotype, or do you need multiple factors to do that? And the result was really quite stunning to us, as shown here on the top of the blood smear, that the sickle mice in the middle have sickle cells and very early red cells, but the crossed mice shown on the right have a completely normal blood smear. And that's because they induce a lot of fetal hemoglobin, as shown on the bottom left. And in FACS plots shown on the right, they have fetal hemoglobin in virtually all the cells. So this told us that removal of BCL11A in the red cell lineage is sufficient to abolish sickle cell disease through reactivation of fetal hemoglobin. And the same would be true of thalassemia. And this is really the basis for the clinical experiments, which I'll come to later. BCL11A is not the only factor. There's a second one, which is called LRF. Or Pokemon or ZBT7A. This is a factor which is about equally potent as BCL11A, for which there's no real human genetics. We can discuss that later if people are interested, but we can not, if one knocks out either factor, one can achieve about 50% fetal hemoglobin in this cell line called UDEP, which is a very useful cell line for studying human hemoglobin regulation. So really, this is the crux of the story. The LCR in fetal liver interacts physically as a loop with the two gamma genes and drives their expression. The beta gene is off. And BCL11A and LRF act to repress the expression of gamma and drive the LCR toward beta to turn on the beta globin gene. And this is really the basis of the switch. Now, in more detail is shown here now with the sequence of the gamma globin gene promoter, which we've had for many years. And what it shows is that BCL11A binds a region where the sequence recognized by BCL11A is TGA-CCA, and that's the site of point mutants, which we'll come to in a second, in hereditary persistence of fetal hemoglobin. LRF binds at about -200 of the promoter, and also there are point mutations associated with hereditary persistence of fetal hemoglobin. And both of these factors interact with the co-repressor NERD complex, which was shown in the middle. And BCL11A actually counteracts NFY, which is the activator for gamma globin expression. And I'll come to that in a summary form later. So what's the mechanism? Well, BCL11A is a large protein, but at its C terminus, there are 3 zinc fingers called 4, 5, and 6. which bind the sequence TGA-CCA, and the structure of the zinc fingers on DNA is depicted below. And the critical feature here is that fingers 4 and 5 are actually the major recognition fingers for this sequence. Finger 6 has really very little contribution to actually DNA specificity. Now, in order to identify where B11A actually binds in the genome, we've used a number of procedures. The conventional ChIP-seq procedure does not work for this protein for probably technical reasons. And the procedure that does work for localization in the genome is this newer procedure called cut and run, which is essentially a method for native localization protein in chromatin using a protein micrococcal nuclease fusion to attract the transcription factors. And this is a very useful experimental system and is being widely used now. We've used a lot of, done a lot with cut and run, and for those who are interested in computational aspects, there's a pipeline that can be used for processing data. But if one looks at the beta globin locus, which is depicted here, this is a confusing slide, but the main point is shown in the pink stripes in the middle, that we could identify that B11A binds to the gamma globin promoter precisely at the sequence TGA-CC, which I showed in the prior slide, and we'll show in more detail. I think one of the most interesting aspects of human genetics is that that since 1985, we've known about point mutations in the gamma promoter, which can affect fetal hemoglobin expression. And these are unusual mutations. Here, these two are called the Greek form of hereditary persistence of fetal hemoglobin. Francis Collins, the director of the NIH, studied one of these when he was a postdoctoral fellow at Yale. And the other one was studied by the late George Stamatianopoulos' lab. And the mutation in Greek HPFH is the G at -117, which is an A in the mutant form. And this leads to about a 10 to 30-fold up mutation increase in gamma expression. So this is an unusual mutation, which leads to an increase in expression. The genetic basis of this was really not understood until it was recognized that B11A binds that sequence TGA-CCA, and the G is precisely the nucleotide that's mutated. And also in a couple of other forms of HPFH, the C is mutated at position -115. And obviously these mutations disrupt the binding of B11A, which is a repressor, and allows the gene to be expressed. And we can rationalize that now with the crystal structure where finger 5 actually contacts that G residue. But one of the things we noticed by cut and run was that if one looks closely at the promoter, B11A is actually found in the distal TGA-CC motif. But there's another motif we call the proximal motif that's only 20 or so nucleotides closer to the start site of the gene. And that was rather curious. Why would B11A bind only one of these two sites? The sites are identical in sequence. And this is what it suggested, was that B11A binds the distal site in collaboration with the NERD complex. And this leads to a repressed or closed chromatin state at the promoter. So what drives gamma globin at the fetal stage? Well, we surmised that it was likely to be NFY, which is a protein that binds the so-called cat box, which is CCAAT. And indeed, NFY binds the cat box in fetal expressing cells, UDEP1, which is shown here. And in this instance, we could actually show by cut and run and very detailed footprinting, I don't have time to discuss here entirely, that NFY actually binds the proximal region, where cat box overlaps TGA-CCA. So in the fetal stage, NFY and coactivators bind TGA-CCAAT, that the sequence CCAAT in the promoter. And in the adult cells, NFY is missing, is displaced, and BCL11A binds the more distal site. So there's a really very intricate gymnastics that goes on at the promoter. And in fact, this occurs with only 21 bases really separating these duplicated motifs. And this is really the basis of the switch between fetal and adult globin. It really focuses only on this region of the gamma promoter primarily. And what BCL11A does is it directly represses at the promoter, probably by physical obstruction, and it recruits the NERD complex to modify the chromatin in that region to close the chromatin, and it displaces the activator NFY. So we think we now understand in pretty good detail the mechanism of the switch. But I sort of skipped over what was the common genetic variation in BCL11A that led us to study the protein in the first instance. And this is really the work of another fellow in the laboratory, Dan Bauer, who's now a faculty member doing extraordinarily well himself. And while Dan was in the laboratory, he focused on where was the genetic variation that really was identified in GWAS. And that genetic variation is located within the second intron of the BCL11A gene, and it overlaps DNase hypersensitive sites, erythroid DNase hypersensitive sites shown in red in the middle. And these sites are not hypersensitive in other tissues, brain or B lymphocytes that also express BCL11A. And as sort of enlarged at the bottom, one can see this region with the sentinel SNPs is interdigitated with erythroid DNase hypersensitive sites. There are 3 of them, plus 62, plus 58, plus 55. There's kilobases for the promoter. And this is a red cell specific regulatory element. It's actually an enhancer that's about 10 or 12 kilobases in size. And we've shown in mouse studies, it's absolutely erythroid specific. And the BCL11A gene requires this enhancer to itself be expressed in erythroid cells, but not other cells in the body. And so we suggested at that time, which was actually 2013, that this enhancer would be a very good target for genetic engineering because it would restrict any of the consequence of genetic manipulation to erythroid cells as opposed to non-erythroid cells. And this is precisely the region that's now being used in gene editing, as I'll come to later. But what Dan also did was use CRISPR-Cas9 to identify a more discrete region of this 10 kilobase enhancer, which we call the Achilles heel, which is shown below. And this is a very discrete region, which is really a GATA transcription factor binding site, which when disrupted impairs the enhancer to about 20 to 30% of its activity. That is, the majority of the activity enhancer can be localized to this one sequence in 10 kilobases. And all one needs to do is make a cut within this region and use non-homologous end joining to sew things back together again. That's enough to cripple a large bit of the enhancer function. And we suggested at that point, which was 2015, that these sequences would be really amenable and favorable for genetic engineering. So this is really the genetics of BCL11A and therapy depicted here, that common genetic variation, which leads to a very small increase in fetal hemoglobin, has a very small effect on hemoglobin expression, and it's due to common variation within the erythroid enhancer. In the next panel, one sees that the common high variation, which is one gets to about 2 or 3%, one has a little less B11A because of specific common variation. But if one deletes the enhancer, one reduces B11A expression to about 1% or less, and one has lots of fetal hemoglobin. And therapeutic targeting, really, one aims to achieve that through genetic engineering or other means to increase fetal hemoglobin. So how can we use this? Well, obviously, one could think of either reducing or destroying B11A activity to increase fetal hemoglobin, and this could be accomplished in somatic cell gene therapy. Now, what would be sort of therapeutics based on that? Well, there are a number of approaches. One could knock the gene out, one could knock it down, one could edit the enhancer or mutate the binding site within the gamma globin promoter itself. In fact, knocking out the gene itself is too toxic for hematopoietic stem cells and B cells, but the knockdown of B11A is feasible, and that's one clinical trial which has been reported and is still ongoing. Another is to edit the erythroid enhancer, which is another clinical trial I'll come to. And lastly, one could mutate the B11A binding site in the gamma globin promoter. There are preclinical studies being done to achieve that, and that will probably be brought to the clinic as well. So B11A is really a quantitative regulator of fetal hemoglobin. It's sort of like a thermostat. You turn B11A down and hemoglobin F goes up. Now, sort of how much do you have to, what's the level that's critical? This is really a very interesting and unfortunate genetic syndrome, which gives us tremendous insight. This is a syndrome of haploinsufficiency for B11A, which is associated with intellectual disability. It's a sort of an autism-like syndrome where individuals have a deletion or point mutation in B11A on one chromosome, the other chromosome is normal. So they have 50% the level of B11A. In fact, when one looks at a number of these individuals, one finds that they have about 13.5, 15% fetal hemoglobin on average. Is a widespread. Some of these patients have as much to 25 to 30% fetal hemoglobin. So in fact, haploinsufficiency is nearly enough to bring hemoglobin F into the therapeutic range. So our estimate is one just needs to maybe reduce B11A a little more than 50% in order to really get to a therapeutic level. So we would guess something on the order of perhaps 60% would be a goal. So one doesn't have to shut it off entirely. One can leave some expression and still get tremendous clinical benefit. So genetic therapy is depicted here, in which one would take, one takes hematopoietic stem and progenitor cells from an individual, modifies these ex vivo in the laboratory, either with lentivirus or gene editing tools. One modifies a fraction of those cells. The goal is to modify as many cells as possible, and then reinfuses those modified cells into a patient who's been preconditioned. That is, the host bone marrow has been destroyed with chemotherapy, usually busulfan. There are many parameters that are important to those experiments clinically that we could discuss later, but I won't go into at this point. But what are the targets then for genetic manipulation? We now have a whole battery of these available. In one can introduce a beta globin gene in sort of traditional gene therapy as one way, but there are various genetic manipulations one could imagine. One is repairing the beta globin gene itself with what's called HDR, or homology-directed repair, correcting the gene, which might be the simplest and most elegant way to fix a genetic disease, but it's the most difficult in terms of the technology. The efficiency is sort of borderline at this point. One can use B11A RNA with a lentivirus and shRNA, and this is being done at Boston Children's Hospital to reduce B11A expression, which reactivates fetal hemoglobin. That's in clinical trial. One could modify the B11A erythroid enhancer, which is being done. One can get non-homologous end joining, reactivate fetal hemoglobin. One can modify the binding site for B11A in the gamma promoter. And one can, in principle, also modify the binding site through base editing rather than gene editing, which is another approach which will be coming to the clinic as well. So there are different genetic strategies, many of which are based on, in some way, impairing B11A function. So in terms of the genetic therapies which are essentially bone marrow transplant, one has to think about the fraction of cells that one can modify in a particular preparation, the efficiency of the modification, the effect of the modification on fetal hemoglobin. You always have to think about off-target or off-cell effects of the modification. Are you going to affect other hematopoietic cells or other genes? The cell dose is very important in the preconditioning regimen. So having a successful clinical trial depends on many parameters in addition to the underlying science. So it really is, each one of these is very important. At Boston Children's Hospital, David Williams has led a trial, which was the first one manipulating B11A, as shown at the top, which is a lentivirus that's designed for expression of target sequences, but only in erythroid cells. And in this case, was introduced a shRNA or microRNA scaffold, which targets B11A. So in other words, this short hairpin RNA reduces B11A expression. So it's really a new form of gene therapy. It's the first use of shRNA in a gene therapy experiment. And the schedule for administration, preparation of cells and administration is shown here. The first patient who received the therapy is shown on the bottom right, with the physician, Erica Ezrick, who's led the clinical part of the trial at Boston Children's Hospital. This patient is now more than 2.5 years post-infusion of the modified cells, and he's perfectly well 2.5 years later. And this is the time course of the patients right after therapy. You can see in the red that the number of F cells, these are cells that contain hemoglobin F, rose to about 75%, which is the plateau level. Here shown almost a year out. His total hemoglobin F is about 25%, which calculates to about 40% F per F cell, which is sufficient to prevent sickling. And his blood smear is shown at the bottom, which is perfectly normal. And this patient is now, as I said, more than 2.5 years out. At this point, it's at 1.5 years, clinically well. As reported in the New England Journal at the end of December, these are additional patients in the trial. And what's remarkable about this trial is that all of the patients have reached a plateau of about 75% F cells, and in each case with about 40% hemoglobin F per F cell. You can ask, why don't they get to 100%? It's probably because not all the stem cells, the long-term repopulating cells, have been transduced. They still have a fraction, about 20% or so, of sickle cells, but they don't seem to produce any clinical problem. The other trial that was reported at the end of December in the New England Journal was performed by a collaboration of CRISPR Therapeutics and Vertex, which involved a disruption of the Achilles heel in the BCL11A enhancer at the GATA site, as I described before. And this is meant to reduce expression of BCL11A only in the erythroid lineage. And this is a summary of a sickle cell patient who was treated, in their case, achieving essentially 99% F cells, and on the bottom, a total hemoglobin F of about 46%. And the thalassemia patients have been treated as well. These are severe beta thalassemia patients who've had a similar outcome. This really summarizes the recent clinical studies, which I think really are a triumph of sort of translation of clinical medicine. The trial on the bottom left is the Vertex CRISPR Therapeutics trial of editing, and on the right is the shRNA trial for sickle cell disease. And as summarized in short commentary in the journal Science, both of these seem to fix the clinical disorders, and the patients are about equally, seem to have an equally good response in both studies. So what's in clinical development now, just as a summary? Globin gene addition is using a lentivirus, sort of a more traditional gene therapy approach, pioneered by Bluebird Bio, is approved in Europe for beta thalassemia. It's still ongoing and is likely to be approved for other applications. There are various other attempts at globin gene addition using either a beta gene or a modified gamma gene. And then there are the studies with lentivirus, shRNA, and gene editing, which were just reported. So I think we can now conclude that that the genetic approach to therapy is validated. That is, all the basic science has been sort of validated in vivo in people, and BCL11A is a therapeutic target that can really likely cure patients with these disorders currently. So, but there's still a problem in that gene therapy is really an expensive, very low throughput approach. So what do we need to do going forward? I think one is to have non-ablative preconditioning, perhaps with repeated administration of modified cells, to make it a less toxic and expensive approach. And then there's still the vision of having an in vivo genetic therapy with new delivery methods via the viruses or gene editing approaches, where one would not need a preconditioning at all, where one would just come into a clinic and get a shot and have your therapy. I think this is very, very far in the future if possible, but it's certainly something that is being thought about by a number of people. What remains to be done? Well, I think if we really want to extend this kind of approach to many individuals and to really lower the burden of disease, we also need better therapeutics or better drugs. And so I think the goal really has to be to find small molecules that could inhibit the critical components of the switch, for example, BCL11A, to actually induce fetal hemoglobin in people in a targeted manner. The advantage of this is one could use population-based therapy that is really high throughput and reach many more individuals. I don't want to suggest this is easy. This is exceptionally challenging. It's what the pharmaceutical industry would call drugging the undruggable, transcription factors. But I think it should be the goal, because this is probably the way to have the greatest effect on disease in the greatest portion of the population. So this really does have to be the goal going forward. As suggested by David Weatherall, that really this is a global problem and we really need to attack it in in a means that would address the global aspects of the disease. So the vision going forward, and summarized at the bottom, would be to achieve pharmacologically what we can now do through genetics. And I think that's maybe the challenge for the future. So the take-home messages are, and I think now the mechanism of fetal to adult globin switching is coming into much clearer focus, and BCL11A is a critical component, perhaps the most critical component. It's a target now for therapy of the hemoglobin disorders. In the clinical trials, both gene therapy and gene editing, based on the scientific discoveries, have validated reactivation of fetal hemoglobin through manipulation of BCL11A as a therapy for sickle cell disease and beta thalassemia. And the present challenges really are expanding the genetic therapies by improving the throughput and developing on-target small molecule therapies to address global aspects of these disorders. And in summary, I just want to say that many, many people have contributed to this work. This is the current laboratory, but there are many who are not shown here have contributed in the past to the studies we've done in our own laboratory. Thanks very much, and I'll be glad to take any questions at this point.
Thank you, Professor Orkin, for taking us through the journey of understanding hemoglobin switching, its regulation, and how it has been applied in clinical trials. Let me start with a question to you about the range of hemoglobin that we see in hereditary persistence of fetal hemoglobin. You mentioned the lower end, but there are patients with much higher levels of fetal hemoglobin. Would there be opportunity to apply some of those mechanisms in treating patients other than manipulating BCL11A?
I'm not quite sure exactly what you're asking.
So some of the, some patients with HPFH have much higher levels of fetal hemoglobin naturally.
Yes, yes.
They could be deletional.
Yes, yes.
So
yes,
yeah, that's
so
what I meant.
I mean, you could delete portions of the beta globin cluster and achieve very high levels of fetal hemoglobin. I think the issue in that comes to really efficiency, in that the larger deletions, the larger the deletion, the lower the efficiency of achieving that deletion. And so whether it would ever be therapeutically practical, I think, is an aspect. The other is the larger deletion, the more chance you have of other genomic rearrangements that may occur. I think there are those who are still worried about the cleavage of DNA as being leading to other genomic events. And in fact, I think disruption, for example, the binding site for BCL11A, if one achieved it on all 4 copies, remember the globin gene, the gamma genes are duplicated, so there are 4 copies. If one did it on all 4 copies, you'd have 100% fetal hemoglobin. So I think there's a lot of room to do better than what's been done thus far. But in fact, going beyond perhaps 40% fetal hemoglobin per cell is probably not needed, except maybe in beta zero thalassemia, you might want a little more, but certainly for sickle cell disease, I'm not sure it's necessary.
So one more question related to that biology. BCL11A obviously has a head start in the understanding of how HPF can be raised, but are you aware of any work being done towards modifying LRF towards clinical applications?
Yeah, I think LRF would be another candidate. I think people are beginning to think of that. Certainly, if you modify the LRF binding site, you might achieve something very similar to modifying the BCL11A binding site. Inactivation of LRF has a greater effect on the development of the red cell lineage, the actual cells, so that impairing LRF may have a cellular effect that's undesirable. One of the things that's remarkable about B11A is that even complete ablation of B11A has really a very modest effect on development of the red cells and the red cell lineage. We saw that in the knockout mouse situation, for example, which is extraordinarily unusual for transcription factors. Usually, an important transcription factor, when knocked out, will severely affect the lineage itself, the differentiation or survival, which is not the case. So B11A is kind of special in that the cell can do away with, it's pretty dispensable for the cell. LRF may be more critical, but certainly if one modifies the binding site for LRF, you would achieve, I think, significant hemoglobin F induction.
Thank you. So we have a question from Dr. Sangam Goswami. How pancellular and heterocellular hemoglobin F regulation works? Can we predict the nature of mutation, impact of the mutation over it?
Again, I'm not quite sure exactly.
Yeah, I'm not sure what exactly.
I think maybe the question relates to the fact that traditionally, particularly hematologists have referred to heterocellular HPFH, where there's tremendous variation in F per cell, versus which is pancellular, where there's a significant amount of F per cell. What's interesting is B11A also was noticed in the GWAS study by Swilethine, had to regulate the number of F cells. So it obviously regulates both the level and the number of F cells. I don't think we understand much about the heterocellular form of HPFH. It has probably more to do with replication kinetics or something of that sort. I think it's not the same as regulating the gamma gene itself. That's about the best I can do there.
Thank you. There's a question from Dr. Seema Shah. Hydroxyurea response can be variable in patients, and is there a way to predict that?
Yeah, that's another great question. I would just summarize to say that we do not understand how hydroxyurea works. The best explanation is it works again by affecting the replication kinetics of the progenitors, which somehow influences hemoglobin F expression. We do not understand how hydroxyurea works. We don't believe it has really any direct impact on gamma gene regulation per cell. We think it's a sort of cellular phenomenon, so it's something separate. And it does not, as far as we can tell, impact the actual intrinsic mechanism of repression.
There's a question about gene therapy now, and whether at this point you are ready to opine whether lentiviral or gene editing is a better approach.
Yes.
I guess.
Yes, I take I take the approach of all shots on goal, we say.
Absolutely.
Which is, I take an agnostic approach that
Yes.
we don't know. I think I would turn it around and say that I think we can say at this point, from the lentiviral and the gene editing, that they both work. And the outcome is probably good enough or near good enough for many patients with either approach. I think what it will come down to in the long term may be more a strategy, a business strategy or an economic strategy. What is the easier strategy to apply in a clinical setting repeatedly? In the case of lentiviruses, you have to make a preparation of lentivirus, of high quality, high titer virus, which is, people tell me, not that easy to do all the time. Whereas in genetic editing, all you need to do is have a good protein preparation and good quality nucleic acid. So it may be that will be easier to implement. Then the question comes down to what are the relative side effects? In one, you have insertional mutagenesis as a theoretical possibility, although lentiviruses have been proven to be quite safe. On the other hand, with editing, one has a question of off-target effects and genome effects. And again, I think it's only going to be experience and practice which will tell us. My guess is that there'll be many different approaches which will all give a satisfactory result, and we'll just have to see what's the easiest to apply to the largest number of individuals, and also to pay for when people actually have to pay for these therapies.
Thank you. There's one more. Dr. Mohan Kumar wants to ask you, why is there no naturally occurring variation in DHS plus 58 GATA motif or BCL11A enhancer, but still editing of this GATA motif shows higher level of HbF induction?
Yes, it's interesting. It's a very astute question. It turns out that the common genetic variation that is in one of the other hypersensitive sites. It's not in the plus 58, which is the one with the largest activity. I sort of say it's what genetic variation is what nature gives you. It's just the luck of the draw. That's what happened to occur. And I think it's sometimes what nature does isn't necessarily what is the most optimal. The other thing is the different hypersensitive sites likely have roles at different times in the sort of life and development of the red cell lineage. So plus 58 is the most important in most of the settings that we've been able to study, but that doesn't really mean the other sites are important at all. And it may be that disrupting more than one element in the enhancer would have a greater effect. And we think it would, and that's work that Dan Bauer is doing and bringing to the clinic as well.
Okay, I think we have exhausted the questions that we had. There was one more about the age at which appropriate time for gene editing in sickle cell disease patients. I don't know if you want to take that question, Professor.
Well, I think sickle cell disease is one of these disorders in which, and the same would be true of thalassemia, when you have damage to an organ, or particularly the brain, let's say, it's cumulative. And the best time to intervene would be the earliest time. So in principle, we'd want to do this in children before they had any major adverse events or accumulated damage. From a clinical perspective, obviously, the trials have to be initiated first in older patients, and when found to be satisfactory, move to younger age groups. And that's been done, for example, the Children's Hospital trial began in adults, and it's now being performed in children of several years of age, and if successful, will move to even younger. So I think the goal would be to do this as young as safe to perform the procedure.
Absolutely, yeah. So looks like there are a few more questions coming in. Great talk, thank you. Could you comment on the status of the approaches that focus on redirecting the enhancer to the beta globin gene, Gerd Blobel's group?
Yeah, so Gerd Blobel, who's one of my former trainees, has shown elegantly that one can basically force a loop between the LCR and a globin gene and drive expression. That could be another form of lentiviral gene therapy. I think from a practical standpoint, it's unlikely to come to clinical practice just because there are already other methods further along that have shown promise. And also, I think the notion of forcing looping between chromosomal elements, elegant and experimental sense, may not be something in a clinical setting one would want to favor.
Yeah.
Might have off-target effects, which would be unforeseen.
Okay, maybe we will close. Shiva, there's one last one, I guess. Considering HbF's oxygen binding curve, is there a threshold percentage above which HbF expression can be physiologically disadvantageous in adults?
Yeah, well, I always say the more fetal hemoglobin, the better. There are very, very rare individuals of 100% fetal hemoglobin through
Absolutely.
very rare deletions. They're perfectly fine. Even the mothers have given birth to babies perfectly fine. I guess the only situation might be if you're if you had 100% fetal hemoglobin and you're some sort of marathon runner or something, and you needed to release more oxygen to the tissues. But I always say the more the better on fetal hemoglobin.
Thank you, Professor Orkin. It was a great talk and wonderful discussions. Thank you very much for your time. I hand this back to Dr. Shivaprakash.
Thank you. Thank you, Professor Orkin, for the very interesting talk. Thank you, Professor Alok, for sharing the session. Thank you so much for both of you.
Okay, it's a pleasure. Pleasure. Be well. Be safe. Yep. Bye-bye.
Bye. Bye-bye. Thank you.